Hello, everyone. I'm going to talk about a fast pipeline that we have developed for species tree inference using placement in hierarchical orthologous groups. Inferring a species tree is a fundamental problem which needs marker genes or orthologous groups. These are defined as groups of genes that emerge from a common ancestral gene through speciation. We can use conventional orthology pipelines to identify marker genes, but these are computationally intensive and are not scalable. Alternatively, we can use pre-computed markers like BOSCO, but it might not be available for all clades. Another drawback is that by increasing the number of species, we will have less universal and single copy orthologs. The standard pipeline of inferring species trees starts with protomes of a species. Here, for example, we have 3 species, and by identifying orthologous marker genes, we have 2 orthologous groups here. In each orthologous group, we have at most 1 protein per species. Since B1 and B2 are paralogs, we choose only one of them, as we can see here. The next step is to run multiple sequence alignment. Once we have the aligned sequences, we can concatenate them based on their species. The last step is to run tree inference methods like IQ-tree or FastTree. When it comes to the runtime, the bottleneck is this step. So the focus of our method is identifying orthologous marker genes. The basis of the proposed pipeline called FastOMA is the fact that we already know for many species what the gene family are and where the duplication happens. We use the OMA database, including 2000 genome, which is a database of subfamily of genes, and we map proteins on them. Traditionally, it was done by finding the closest sequence using methods like BLAST or diamond. The issue is that the closest sequence might belong to a different subfamily. As an example, here we draw a gene tree, which at this point, the duplication happened. The correct subfamily is this one. We can see the query protein here. But when we run those methods, we will find this subfamily. This is the closest one, but it's wrong. This happened because of asymmetric evolutionary rates. Our solution is the OMAmer tool. It is a classifier based on subfamily-informed k-mers. In order to describe our accelerated pipeline, which is FastOMA, we start with the protomes of a species. We use OMAmer and map each protein onto a subfamily. Here, for example, protein A is mapped. In this subfamily, protein B1 and B2 are mapped to the same subfamily. In order to avoid paralogs, we remove these proteins. On this subfamily, we have protein E and C from 2 different species, and we keep them. The next step is to find the best orthologous group for each subfamily. Then, using the proteins of this orthologous group from the database, we combine with the query protein and we run the multiple sequence alignment. Then we concatenate them and infer the tree. In order to evaluate our method and pipeline, we use the BERT 10,000 genome project. The second phase, which was published recently, includes around more than 300 BERTs. When we run the standard pipeline and our pipeline, we can see that a standard pipeline needs around 2000 CPU hours. but FastOMA needs only about 50 hours. The rest of the pipeline is quite similar. So we can see that FastOMA is about 50 times faster than the standard pipeline. Here we can see the tree that was depicted using phylogeny website. Here the dark blue shows good consistency between these two trees. In order to quantify these inconsistency, we used Robinson folds metric. There are 69 RF partitions that exist only in a standard pipeline, but we couldn't find them in FastOMA out of 700 partitions. We also consider NCBI taxonomy. We know that NCBI taxonomy is not fully resolved. When we compare this NCBI taxonomy to the tree that was provided by the BERT 10,000 Genome Project, we can see a relatively small overlap in terms of Robinson folds partitions between these two. We also compare the tree that we inferred with these two trees. Here we can see that there are 113 partitions which are in common between BERT 10K and FastOMA, which shows that FastOMA is more consistent with the BERT 10K paper. In conclusion, FastOMA is a reliable and scalable solution for the tree inference pipeline. Thanks for your attention.